All right, it gives me great pleasure to introduce our speaker today. It is Professor Yuri Saalmann, who comes from the University of Wisconsin-Madison. He's associate professor there. He's also a scientist at Wisconsin National Climate Research Center. Yuri has a PhD degree from Australian National University, and then he had done postdoc work in two places. One is the Royal Holloway University in London, and the other one is Princeton. 5 years in Princeton. Yep. And so his main research is investigating neural bases of cognitive control, and the program is directed to understanding information processing in large-scale brain networks in healthy subjects and subjects with psychiatric disorders. Today we will hear an overview of neural bases of consciousness. Thank you, Yuri.
Thank you for that. Yeah, thank you for that kind introduction, Peter. So the reason a neuroscientist like me is giving a talk to the Department of Electrical and Computer Engineering is convergence. So as you may know, one of the 10 big ideas from the National Science Foundation is growing convergence research. The basic premise here is that big problems in science require many different approaches from multidisciplinary teams. And so this brought together Peter and Seema and Chuck to study consciousness. And I must say this has affected me in profound ways. One of them is that, when I used to go to sleep at night, I used to dream about lying on a beach in Australia where I grew up. And now I dream about Gaussian processes. I guess I should be thinking better for this.
You are very conscious.
Exactly. So in the spirit of convergence, today I thought I'd give you a general overview of the neural basis of consciousness, but with a focus on the types of problems that we're all working on, which will include causally manipulating consciousness using deep brain stimulation. So if there's any questions that you might have, feel free at any moment just to chime in, and hopefully we can sort of bring us all close together onto the same page. So the problem really that we're working on is identifying the neural correlates of consciousness, all right? And the neural correlates of consciousness just means the minimal brain activity that's necessary and sufficient for consciousness. And if you ask any neuroscientist, they'll agree, yes, the entire brain is necessary for consciousness, but not all parts seem to be necessary. So for example, if we go to the back of the brain here, you can see this region known as the cerebellum. Now, the cerebellum is actually involved in fine motor control amongst other functions. But if you damage the entire cerebellum, consciousness persists. So it seems like the cerebellum is not necessary for consciousness. And on this slide, I've highlighted a number of different brain regions that people have proposed to be important for consciousness. But what I really want to do now is just focus on the actual surface of the brain here, which is known as the cerebral cortex, all right? And we're going to focus on the frontal lobe, as you can see here, which has expanded greatly over the course of mammalian evolution. So you might think, well, if it's really expanded to its largest sort of portion here in humans, maybe it plays a vital role in consciousness. The only sort of hiccup here is that if you have damage to the actual frontal lobe, you don't always get disturbances in consciousness. So that has led to a number of other researchers proposing that maybe more posterior parts of the brain, such as the parietal lobe, the occipital lobe, and the temporal lobe, may contribute more to supporting consciousness, all right? And this is really the underpinning of the so-called front versus back debate that's prominent in the consciousness science field. So basically, what are the differential roles of the front of the brain and the back of the brain in supporting consciousness? So this maps onto a number of major theories of consciousness, which I'll just briefly review here. And so what you can see on this particular slide is a number of lateral views of macaque monkey brain, all right? And if you look at the brains on the left, we're showing the mechanisms underlying two of the major theories of consciousness that are really pushing for the frontal lobe as playing a vital role. So if you look at the top left here, you can see a theory known as global neuronal workspace theory. And here the idea is that information from our sensory environment is first transmitted to the back of the brain here, in this case, visual cortex. And this information about our sensory environment is then transmitted via these black arrows, so-called feed-forward pathways, to the frontal lobe shown here in pink. And the idea is that if you're going to consciously perceive a stimulus in the environment, you'll actually need a large increase in activity in this region around 300 milliseconds after the onset of that sensory stimulus. And then this increase in activity then spreads via these red arrows, these so-called feedback pathways, to be broadcast across the rest of the brain. In a somewhat related fashion down below left, you can see what's known as a higher order theory. And here, as before, sensory information from the world around us is transmitted to the back of the brain here. So we have a first order representation of our sensory world. However, the proponents of this theory says that's not enough to support consciousness. You also need a higher order representation, for example, in the frontal lobe, a meta representation of this sensory experience, all right? Now, in contrast to these two different theories on the left here, we have on the right two other theories that are now proposing a more prominent role for the back of the brain. And the first at top right is known as integrated information theory. And here the idea is that causal interactions between groups of neurons in parts of the brain, such as the parietal lobe, the temporal lobe, and the occipital lobe, are actually vital for consciousness. And the proponents of these theories, are suggesting, yes, it's the back of the brain that's really important for consciousness. And then finally, down at bottom right, we have known as a first order theory, and this is recurrent processing theory. Here, the idea is that reciprocal interactions, of the sensory pathways in the back of the brain are sufficient for consciousness, all right? So we can actually test these different theories by, for example, looking at the role of the frontal lobe in sensory processing. And one way we can do this is by first exploiting a phenomenon known as binocular rivalry. So if you're not familiar with this particular paradigm, the idea is we're going to present two different stimuli to each eye, right? And you can do this by having a divider between the eyes or by using specialized goggle systems to present the stimuli. So in the example here, what you can see is that we're going to present green bars moving to the left eye, and we're going to have red bars moving to the right, presented to the right eye. And what subjects actually perceive is that sometimes you're only sort of experiencing the green bars moving to the left, and sometimes only experiencing the red bars moving to the right. And we can get insight into what the actual subject's experiencing because we can ask them to press buttons on a response match, for example. Left button when sensing leftward movement, right button when you're experiencing right movement, right? But in addition to this voluntary report, we can also have an automatic readout of what subjects are experiencing from their eye movements. So when subjects are actually experiencing leftward movement, the eyes take a particular tracking pattern as shown here. And in contrast, if you're experiencing rightward movement only, the eyes track the stimulus in a different way, all right? So now using this behavioral paradigm, we can take subjects into an MRI scanner where we can actually monitor their brain activation, right? Whilst these actual perceptual experiences are switching. So what you can see presented here in the middle of the slide is one hemisphere of the human brain. We're looking at it from the front. And these yellow-red areas are the parts of the brain that are activated during these perceptual experiences here. So the first thing I want you to know is that we have activations across different parts of the brain, including the frontal lobe here and more posterior areas like the parietal lobe and the occipital lobe at the back of the brain. But what's an issue here when we're interpreting this data is that the frontal lobe, as shown here in the black circle, is actually involved in the planning and execution of voluntary movements, all right? So it's not clear if this activation is due to the voluntary report, the actual actions, or if it's due to consciousness per se. So the experimenters actually repeated this experiment, but no longer requiring a left or right button press to signal what they're experiencing. And instead, they actually used this automatic eye movements to get an insight into what the subject is experiencing. And what you'll notice now is that without the voluntary report, you no longer get the frontal lobe activation that you had previously, but you still get the activations in more posterior brain areas like the parietal lobe and the occipital lobe, all right? So these type of experiments, at least this particular one, suggest, well, maybe the frontal lobe needs to be questioned in terms of what role it's really playing in conscious perception. And so I want to show you a different way that we can probe which brain areas are contributing to consciousness, and that's using electrical stimulation, right? So In severe cases of epilepsy, for example, part of the clinical diagnosis, as Chuck will tell you, is that you can actually apply small electric currents to various parts of the brain. And there's no pain receptors in the brain, so you can actually perform these particular procedures without the subject actually feeling, right? So we're going to see an actual video of a case study while we're actually stimulating here in the back of the brain, the temporal, right? So I'll start the video now. All right, we're having a problem with Zoom. So we didn't actually try it with Zoom. Good, so that's my fault. Switch back. Oh, there we go.
Yeah, it's almost me.
It should actually. So there's no current applied in the baseline control. All right, so you get the basic idea that if we stimulate, the temporal lobe, go back to the actual laser pointer. All right, so when we stimulate the temporal lobe here at the back of the brain, correcting myself, you can actually influence what subjects are actually perceiving. You can similarly influence perception from stimulations of the occipital lobe as well as the parietal lobe, but it seems to be less easy to actually influence perception when you stimulate the frontal lobe, at least from the data that I've seen. Okay, so what I'd like to do now is actually focus on the pattern of electrical activity in some of these brain regions that we've been discussing. Right? So what we're going to do here is look at an EEG experiment. So EEG is a non-invasive method of monitoring brain activity. So you have electrodes on the scalp, and these electrodes will pick up the activity from the underlying brain tissue. All right. And what we're going to do is we're actually going to try to perturb the actual brain activity using another type of non-invasive method called transcranial magnetic stimulation, or TMS short. The idea is that you're going to induce magnetic fields, which will influence the electric currents in the underlying tissue. All right. So what you can see here up top are the actual recordings of brain data from the EEG electrodes. And we're going to actually perturb the actual brain using transcranial magnetic stimulation at this point here. All right. And what you can see is that after we perturb the brain and now the subject's in the wake state, you can see that we're influencing the activity, right? There's a significant change above baseline. And these red curves show you where the significant changes happen. So what you'll notice is that immediately after the stimulation of the brain, you get these changes in activity across different EEG electrodes that reverberates or persists for up to 300 milliseconds or more. And we also have, if you look down below, a spatial spread of activations of the brain in the wake state. So you can see here in the bottom row, overhead views of the brain, and the yellow-red regions here are showing which parts of the brain are currently activated. So we're going to look at how the brain's activity sort of evolves across time, moving from left to right. So what you'll see initially, we use this TMS method to stimulate the premotor cortex, and this initial activation spreads to the opposite hemisphere, as you can see here, and then starts moving towards the back of the brain, and then eventually sort of echoes back up to more anterior regions again. All right, so we have this temporal and spatial dynamics occurring in the wake state. But what we're going to do now is contrast this with a reduced conscious state, such as that during non-REM sleep. So same experimental format as before, but now you can see when we stimulate the brain using TMS, we get an initial significant change in activity, but this soon peters out, right? It's over by about 150 milliseconds. It's back to baseline. And we do not get the same spread of activation across the brain. So you can see here, here's our initial stimulation side, and it stays rather localized across time, all right? So you have greater complexity of brain activity in the wake state compared to reduced conscious states. So we can actually quantify this complexity using, for example, a number of information theoretic measures. And I want to, in this sort of time now, just briefly provide you with an intuition for one of the information theoretic measures that's used in consciousness science. That's integrated information, or phi. And then after that intuition, then I'll show you how we calculate it. So if you look down to the bottom left of this slide here, you can see a number of black circles. Each of these circles might represent a node of a brain network. So each circle might be a different part of the brain. And you'll notice that all the actual nodes within this red circle have causes and effects on each other, shown by these black arrows. And that's very different to some of these other nodes outside the actual red circle. And so the intuition for integrated information, or phi star, is that any system that has these integrated causes and effects can generate this integrated information concept of phi star, right? And so translate that into more concrete terms of the brain. If you look up the top here, we have a lateral view of the macaque monkey brain. We've color-coded here the frontal areas in blue, the parietal areas in the reddish colors, and then some deep brain areas that we'll talk more about in a moment, the caudate nucleus in green and the thalamus in purple, right? And so you could imagine here that these particular areas can have causes and effects on each other, similar to our toy network down below, through these various anatomical connections here that I've shown you with the black arrows. So you could imagine a subsystem, down below, where the parietal cortex in red, the caudate in green, and the thalamus in purple, have these causes and effects on each other to generate what I'm calling integrated information. All right. So with that intuition, let's think about, in more concrete terms, how we calculate it. So the first step is actually to calculate the mutual information of this particular system, right? And we can use whatever electrical activity that we have at our disposal. And we'll talk more about those signals in a moment, all right? So calculating the mutual information allows us to sort of capture the mutual dependencies between these different brain parts. And then the second step is actually to calculate the mutual information of the partition system. So the basic idea is we're going to artificially cut this system and see which cut leads to the least amount of information. And we call this the minimum information partition, shown here by the red dashed line. And the idea is this really gives us some insight into which parts of the brain are more integrated, right, or on the same side of that partition. And then the final step for calculating integrated information, or phi star, is simply to take the mutual information of the entire intact system and subtract the actual mutual information of this partition system, all right? And here you can see a postdoc in our lab, Mohsen Afrasiabi, who was the primary driving force behind this computational work. And so what we actually did here is record from all of those brain areas I mentioned before, and we recorded a type of neural signal known as a local field potential, LFP for short. And you can see a time series of this local field potential here. And this local field potential reflects the pooling of activity of maybe hundreds of neurons in each of these individual brain areas, all right? And so what we then did is calculate the power, spectral power density of these local field potentials, LFPs, and then a number of information theoretic measures, including the entropy, or H. So that gives us an idea of how many different states our system can be in. We then looked at the mutual information, and as well as this integrated information, or phi star concept that I introduced on the previous slide. So we took these 4 different neural measures and then fed them in to support vector machines to see if we could actually classify our 3 different states of consciousness here. So we wanted to see if we can take activity from the wake state, sleep state, and anesthetized state and classify it appropriately. So what you can see on the plot at top right is our classification accuracy, and you'll see that we have a chance performance of 33%, because we have 3 different conditions that we want to classify. And you'll notice that all of our different classifiers performed above chance. However, the information theoretic measure, integrated information or PHI-STAR, and the local field potential power gave us the best result. So we'll focus a little bit on those now. So what we're going to do now is interrogate this data and basically ask which brain areas contributed most to this classification performance. And so to do this, we actually measured something known as the mean drop in accuracy, or MDA for short. And the basic idea here is we're going to repeat those classification analyses, but now leaving out each brain area in turn. And so the idea is if you leave out a brain area and there's a large drop in the classification accuracy, really that brain area is making a significant contribution to consciousness, right? Or at least the classification of these conscious states. So here on this graph, wherever you see higher bars, it means those brain areas are contributing more to our decoding of that conscious state. So what you'll see is that these colors map onto the figure above, that the thalamus deep in the brain in purple, another deep brain area, the caudate nucleus in green, and this parietal cortex area in red towards the back of the brain all contributed most to decoding these different conscious states. And these were the support vector machines using the local field potential power, or LFP power as inputs. And similarly, if we actually go to the support vector machine using the PHI-STAR integrated information measure as the input, you'll see once again that it's our deep brain areas in green and purple, as well as the parietal cortex in the red colors that contributed most to decoding our different conscious states. But what you'll also notice is that no matter what classifier we use, the frontal cortex here in blue in the front of the brain contributed relatively little, all right? So this does raise questions about what role the frontal lobe really has in supporting consciousness, okay? So what I'd like to do now is go to a much higher spatial resolution and record the activity of individual brain cells across different conscious states, right? And so what you can see up the top here is actually a lateral view of the macaque monkey brain. And we're going to record from both the front of the brain, known as the frontal eye fields of FEF here, and more posterior or back part of the brain, the lateral interparietal area or LIP. All right. Now, these areas on the surface of the brain are actually only about 2 to 3 millimeters thick, all right, which you can see in the inset here. And they're organized into 6 different layers. And usually we classify the superficial layers as layers 1 to 3, the middle layer is layer 4, and the deep layers 5 and 6. And as we'll see in coming slides, these have different functional roles in brain processing. And then we use what's known as linear electrode arrays, right, which are implanted across all these layers of the actual brain areas. So these individual little circles here are individual electrode contacts, about 12.5 microns in diameter, which allow us to record the activity of individual brain cells, all right? So the actual former graduate student in my lab, Michelle Redenbaugh, was the primary driving force behind these recordings. So what we're going to see now to the right is an actual outcome of these recordings. So we have in the left column the frontal lobe recordings, FEF. In the right column, we have the parietal lobe recordings, LIP. And then each of the rows correspond to our different layers of these different areas, all right? Now, we've color-coded the different conscious states. So the activity in the anesthetized state is shown in blue, in the light sleep state in teal, and the actual wakeful state in the orange color, all right? And then as our measure of neural activity, we've got the spike rate, all right? And we'll talk more about this in a moment, but individual neurons generate electrical pulses, which we call spikes, which actually transmit information to other neurons, all right? So the greater the spike rate, the more active these neurons are. So what you can see is that the superficial layers of our cerebral cortex on the surface of the brain doesn't really change its activity much in our paradigm across wakefulness, sleep, and anesthesia. And there's no significant difference either in the middle layers. But where we see the significant differences are in the deep layers of these different areas. So you can see we have the highest activity in the wake state here, less activity in the sleep state, and least in the deeper anesthesia state. All right. So this suggests that the deep layers of the cerebral cortex are important for consciousness. All right. So what I'd like to do now is go to an even higher spatial resolution and see how information is transmitted along these different cells across different conscious states. And so what I'm going to show you here first is one of the methods that was necessary to manipulate these individual cells at a fine grain level. And this method is known as optogenetics, all right? So this basically exploits the fact that in nature, like in the algae pictured up top left, there are these light-sensitive proteins. So if we zoom in on one of these light-sensitive proteins here, you can see that down below. So the protein here is in blue. And basically, it's got a central pore made from this protein. So normally it's closed. However, if it absorbs the energy from blue light, then this actual central pore channel opens up, and that allows positive ions, for example, to flow through the channel. So this basically will induce electric currents into the cell, which can then change its electrical properties. So you can imagine then that we can take the genes encoding these light-sensitive proteins. We can introduce them into viruses. So they act as viral vectors to actually infect different cells in the brain in a non-pathological way. And then those particular cells will have the DNA or the genes for this particular light-sensitive protein. And then they'll express or manufacture the protein so it's on the surface of the cells. So what you can do next is then insert very fine optic fibers into the brain, and using a weak blue light laser in this case, shine light down into the brain to selectively activate just those brain cells that have these light-sensitive proteins in the actual cell wall or membrane. All right, so we can now switch neurons or brain cells on and off just by switching off this blue laser. Okay, so armed with this technique, we now go to the main experiment that I want to highlight. All right. So what you can see here on the actual left is the cerebral cortex, right? The surface of the brain. So this is like the top 2 to 3 millimeters of your brain. And you can see those different layers that I mentioned before, superficial, middle, and deep layers. So what we're going to focus on here now are cells that have their, cell bodies, as they're called, in the deep layers. The ones that we identified in our previous monkey work that were actually seemingly changing their activity across different states of consciousness. So these green particular cells are known as pyramidal cells or pyramidal neurons because they have a pyramidal or triangular cell body. And what I want to highlight are 2 different parts of these cells. All right, so these extensions that go towards the surface of the brain here are known as dendrites. And these dendrites represent the input to the neuron. So other neurons or brain cells in the brain are actually going to communicate with this cell via these dendrites. Okay, and the other part of this cell that I want to highlight is the actual cell body or the soma here in the deep layers, all right? And the soma here represents part of the cell that's close to the output. So we talked about these cells generating electrical pulses and other spikes. Well, in the vicinity of the cell body here, the cell is going to generate pulses which will transmit to other neurons to communicate with a broader network of brain tissue, right? So what we can do in these experiments is actually, now that we've utilized this optogenetic technique, here's our optic fiber. We're shining in the blue laser light through the optic fiber. We've got our light-sensitive proteins here on the dendrites. We can now switch on the laser to activate the inputs to the cell. And we're going to measure the actual electrical activity at these dendrites, the inputs to the cell with an electrode, as well as the cell body or soma here with another electrode to look at the output. So we can monitor the input-output relationship of the brain cell, right? So we can see how information is transmitted along this particular dendrites across the cell. Okay. So if we now look to the figure on the right here, what you can see first up the top is the activity at the dendrites, right? The input to the cell. And so what you'll see is in response to this optogenetic activation of the dendrites, we have with this red curve a large change in the electrical activity in the wake state in the mice here. And you'll notice now for the black curve, the case when the mouse is anesthetized, so in the reduced state of consciousness. And whilst there's a modest reduction here, there's still a significant departure from the baseline state. So it suggests that the inputs to the actual neurons or brain cells here, the deep layers, are still functional, right? So the cells can still receive information from other neurons. So what's breaking down in anesthesia? That's the big question here at the level of the cell. So to answer this, we now have to go down to the cell bodies or soma here. And if you look at the red curve, this is the actual response from the soma in the wake state. And you can see a very large response during the wake state. But in contrast, if you look at the black curve when the animals are anesthetized, you've got a very little change in the activity in the soma. So this suggests that information transmission along this particular dendrites here of individual neurons is breaking down in the anesthetized state, likely in other reduced conscious states, all right? So we fundamentally altered the input-output relationship of this brain cell during general anesthesia when subjects experiencing reduced consciousness.
All right, so Yuri, sorry.
Please, yeah.
What has happened during anesthesia to these bats? Why are they not operating the way they operate under awake condition?
That's a great question. And I think you'll love the answer. The reason why I say that is part of the project that Peter Seam and Chuck Paul are working on is interactions between the deep brain areas like the thalamus and these cells in the cortex. And it turns out that the cells in the deep brain area of the thalamus actually send anatomical connections to this region of the cell here. And when you shut down the thalamus in anesthesia, you shut down the activity of these inputs, which reduces the actual excitability of these cells, which seems to break down these particular transmission along this pathway. So the deep brain areas of thalamus allow the inputs and outputs of these cells on the surface of the brain to be coupled. And then during anesthesia, because you reduce that thalamic input, which is regulating the electrical activity across this fiber here, you decouple the input from the output. So
that means in another word, that the anesthesia is working.
Yes, but we now know how it works in a fine controlled way. And the reason why that's important, I mean, it's important philosophically and scientifically to understand the neural correlates of consciousness, but clinically, 20 to 40,000 people alone, and sorry if anyone's going for surgery in the next few days, talk to Chuck, I'm sure he'll tell you all will be fine, but show some awareness under anesthesia. So 20 to 40,000 people each year in the US, right? And a fraction of those might actually remember those experiences, and that can lead to post-traumatic stress disorder. So having a better understanding of the neural mechanisms of anesthesia can not only help us scientifically for answering the big questions of what is consciousness, but also to improve the clinical situation. All right. And so there are many different drugs with different chemical targets or molecular targets. It seems that all of them seem to affect this particular process. So now maybe we can design better drugs to disrupt consciousness, but at the same time not disrupt cardiovascular processing or respiratory processing, right? And maybe separately manipulate pain sort of perception. So that's why that's really important for people like me. Yeah. All right, so we can actually visualize these cells in real time in the deep layers, especially in layer 5, right? And so you don't just have to see the plots and take my word for it. So what we're going to use now is a particular type of method known as calcium imaging. So this uses calcium indicators that fluoresce. So the greater the calcium concentration inside the cell, the greater fluorescence that we can pick up with special microscopes, all right? So the idea is that the greater the calcium, the greater fluorescence, the more electrical activity of these cells, all right? So I'm going to try to actually go off screen and test all my coordination levels. Can I call upon your miraculous abilities? As you can see, I'm the PI, I don't do anything. All right, so what you're seeing here, these yellow reddish blobs, so to speak, are individual brain cells in those deep layers of the cerebral cortex. So the more you see these yellowish sort of activations, changes in fluorescence, the more active these cells are. So what you're seeing here is the activity of these deep layer neurons that we just saw that were actually being decoupled from inputs and outputs in an anesthetized state directly, all right? So this is in the wake state now. So you see that there are many different neurons are active, they're active tonically, they're firing or changing their activity quite dynamically across the time in the wake state, all right?
Is this real time?
I think this has been sped up twice, perhaps, but I'm
2x.
yeah, 2 times, yeah, perfect. It's going to struggle because I can't see the counter. All right, and now we're going to see the actual, same neurons, but in these deep layers of the cerebral cortex on the surface of the brain, but when the animal's in ether time. with a particular drug known as isoflurane, which is commonly used clinically. All right. Just so this. Hey, fantastic. All right, here we go. So what you'll see now is a very different pattern of activity. So now there's these hypersynchronized moments followed by long pauses. Hypersynchronized, long pauses. You can even get much more, hypersynchrony here, and then once again, a long pause in the electrical activity of these cells. All right. So it's a very different pattern of activity that we see in the anesthetized state. Right. These long pauses are really disrupting, the continued processing of information in the cerebral cortex. So the question now is, why do these deep layer neurons actually have such a big influence on consciousness? And so now we're going to have to delve a little bit deeper into the brain circuits that are proposed to be critical for consciousness. And so I'm just going to bring up the laser pointer once again. And so what you can see here at the bottom left of this slide is a medial view of the human brain. And so information from the eyes that would typically be here, obviously, is first sent to the back of the brain, what's known as the visual cortex here. And then it can be transmitted to higher order parts of the brain, like the parietal lobe that we were talking about before. Now, I've schematized the relationships between these areas, as you can see up top on the slide. So what we have is the visual cortex at the back of the brain here on the left. We have the parietal cortex here on the right. And we have those different layers, superficial and middle and deep layers, the top 2 to 3 millimeters of the surface of your brain plotted. And then I'm going to introduce later a central thalamus here, this red region deep in the brain there. All right. So now, the first circuit I want to highlight is actually known as a feed-forward pathway that takes information from the visual cortex to the parietal cortex along these green arrows. And you might well ask, well, what information is this carrying? Well, this is carrying the information from your sensory environment. So every time that you're viewing this particular talk, for example, information is going to be sent via these green arrows from visual cortex to parietal cortex on the brain. Now, these originate in the superficial layers of one area and terminate in the superficial and middle areas of it. All right. We also have what's known as feedback pathways shown here in brown, which are carrying your attentional priorities, your goals. So every time you're focused on carrying out a task, you've got activity being spread across these feedback pathways in brown. All right. And these originate in the deep layers and terminate in the superficial and deep layers of another brain. We also have what's known as these intracolumnar projections, which are shown here in blue. And these allow the different layers of the cortex to communicate with each other. And then finally, I want to highlight the connections between the thalamus deep in the brain. And this is connections that are unique to the central thalamus here. They have reciprocal connections shown in red with the deep layers of the cerebral cortex, and then also connections with the superficial layers. So the reason why I might suggest that deep layer neurons, like layer 5, are so important for consciousness is that the deep layers shown here in blue are involved in nearly all of the key brain circuits that people have proposed as being critical for consciousness. So if you actually perturb activity in these deep layers, you're going to perturb these feedback pathways shown here in brown that are carrying your attentional priorities and goals. You're going to actually shut down some of these connections with the thalamus and eventually the connections with the superficial layers of the cortex, which normally increase the excitability of neurons or brain cells in the superficial layers. And you're also going to shut down these intracolumnar connections, which actually influence excitability of the cells in the superficial layers. And if you do this, eventually you'll reduce the activity in these feed-forward pathways that are carrying the sensory information about the world around you. So if you disrupt these brain feed-forward pathways, you're going to disconnect yourself from the environment. All right. So the question is then, how can we intervene in this system to causally manipulate consciousness? One, you're engineers, and if we don't build it or purposely manipulate it, we really don't understand how it works. And secondly, clinically, we might want to actually manipulate consciousness to treat disorders of consciousness like coma, right? So you might say to yourself, well, the deep layers like layer 5 might be critically involved. However, it may not be feasible to manipulate all these particular layer 5 neurons because, as you've probably imagined, the layer 5 spreads across the entire cerebral cortex. And so to implant electrodes all across the brain would be a challenging exercise. So what are our other options? Well, what I want to propose here is the central thalamus, so deep in the brain, is actually a good candidate for manipulating this entire system via these red arrows as shown here, which can influence all those key brain circuits that I've just introduced. And if you look on the right here, you can see a picture of the human brain. I've cut away the surface of the brain to show you the connections between the central thalamus deep in the brain with the entire cerebral cortex. So the anatomical connections, which would allow the central thalamus to influence all those deep layer neurons, for example, and other brain circuits are shown by these colored lines, all right? So what we're going to do now is try to translate this into maybe more clinical goals to give you an understanding of why I think this research is important for improving therapeutics. So what we're going to do now is look at a number of different states of consciousness now plotted along two different axes which are useful clinically. So we have on the x-axis the level of arousal, the level of vigilance, which you can gauge by just seeing if the subject's eyes are open or closed. And on the y-axis, we've got the level of awareness, the richness of your perceptual experience. And we can gauge this by listening to the response of patients to questions like, do birds fly south for the winter? Yeah. Or do I get mugged at LaGuardia when I first arrived? Okay. And so what you'll notice that top right here, we have conscious wakefulness, which I hope you're still in, with high levels of awareness and arousal. And later tonight, you'll go into a state of sleep where, non-REM sleep, you have lower levels of awareness, arousal. And unfortunately, as I alluded to before, you have disorders of consciousness that might, occur because of traumatic brain injury, and Chuck can talk much more expertly about this than I can, such as coma with very low levels of awareness and arousal, all right? But we also have other disorders of consciousness like unresponsive wakefulness and minimally conscious state, which have impaired awareness. So a goal clinically might be to actually manipulate our brain state to move us from the minimally conscious condition to a state that's closer to the conscious wakefulness, all right? And one way that we might be able to do this is using deep brain stimulation. So we already talked about the fact that maybe the central thalamus, which is located here deep in the brain, right, could be a key target for such a manipulation, right? So we can implant these electrodes, again, no pain receptors in the brain, so it's just to feel it, and then apply electric currents to the actual central thalamus, and part of it is known as the intralaminar thalamus, or ILM. So sorry for the jargon here. But if you stimulate this particular part of the brain, you would increase the activity of neurons here, which would then cause activity to spread along some of those anatomical pathways I showed you on the previous slide, as shown here in red, to change the excitability of these neurons in the cerebral cortex, all right? And so this was actually done in a case study a few years ago, all right? So what we have here is a patient who was in a minimally conscious state for 6 years prior to this clinical intervention. If you look on the left, you can see their image of their brain, and these dark trajectories here show the electrodes that have been implanted. You can see we're going to stimulate in both hemispheres in the central thalamus here deep in the brain. And when the clinicians did this, the actual subject's behavioral responsiveness improved markedly, right? And we can see that in graphical form, right? So what I'm going to show you is the magnitude of the effects here on the y-axis. And first, you can see prior to actually stimulating the central thalamus, there was low levels across many of these indicators. However, when we actually stimulated the central thalamus, you can see that the arousal went up, as shown by this blue bar. We have an increase in the actual communication of the subject shown here. This included the subject who, before the intervention, couldn't respond to questions, but afterwards could actually respond in complete sentences. We have improvements in motor function, which included the ability for them to feed themselves, etc. All right. So this just, basically gives support to the feasibility of manipulating the central thalamus to manipulate consciousness, at least in disorders of consciousness, like the minimally conscious state. So at this point, we can actually turn to animal models to actually better understand the underlying neural mechanisms of why that worked, but also to see if we can actually optimize the manipulation for even better effects. And so what I'm going to show you here at top left, we have the picture of the macaque brain again. Deep in the center is the thalamus, and we're going to zoom in on the central thalamus. For orientation, here's the midline of the brain. This is the lateral ventricle for any neuroscientist here. And this area here in gold is part of the central thalamus, a particular part known as the central lateral nucleus, or CL. all right? So we can actually, target these electrodes using high resolution MRI techniques, for example, and you can see our electrode in situ, in the central lateral thalamus, which is located, if I can bring my cursor back, to, between the medial dorsal thalamus MD and the ventral posterior lateral VPL. All right, so the first thing that we wanted to do was characterize the activity of the actual neurons in the actual, oops, we've gone too far, in the actual conscious state first, all right? So you can see here at top right, a raw recording trace of the voltages recorded from neurons in this central thalamus area, right? So to help orient you, these sharp deflections of voltage here, those electrical pulses that I mentioned called spikes, that brain cells use to communicate with each other. And in the central thalamus, there's a subset of brain cells that actually generate these electrical pulses or spikes at 50 hertz, all right? And interestingly, when you go to reduced conscious states, the actual activity cells drops markedly down to around 10 hertz in some cases. So keep that in mind, we'll return to that in a moment. So what we thought we might want to do with our deep brain stimulation intervention is try to actually mimic the activity of these brain cells in the central thalamus during the wake state to try to increase the level of consciousness. And so that's why we actually stimulated at 50 hertz, applying electric currents 50 times a second to mimic the electrical pulses basically generated by individual neurons in the central thalamus. So our model, and we can argue about just how the validity of this model, but of disorders of consciousness, is to anesthetize our macaque monkeys, all right? So we're in their induced state of consciousness and see if we can override the system, right? By stimulating at 50 hertz to actually wake up the monkey despite, continuously administering anesthetic drugs. And that's exactly what we did. So what you can see here on the x-axis is time across a typical experiment. So throughout the entire procedure, the animal receiving a constant rate infusion of an anesthetic known as propofol, which is widely used clinically and for surgical anesthesia. And on the left here, we have on the y-axis a number of behavioral and neural indicators. So not surprisingly, in the actual anesthetized state, the subject's eyes are closed, there's little muscle activity in the EMG, and you've got these large slow waves, as you can see here, large amplitude waves in the EEG, but also our intracranial sites, in the frontal lobe and in the parietal lobe in the back of the brain. And these large slow waves are thought to disrupt activity, right? It's a hypersynchronous activity of neurons followed by long pauses. And then in this actual pink shaded region, we switched on the 50 hertz stimulation of the central thalamus. And within seconds of actually stimulating, the animal woke up. So you can see the eyes opened, the muscle activity in the EMG increased, and we've got a reduction in these slow waves in the EEG and in our intracranial recording sites. And more than that, the animal started reaching to objects in the environment in a room which they had never seen before. So that clearly sort of suggests that they're perceiving the actual objects and then organizing reaches, as well as vital sign changes and other indicators. And then within seconds of turning off the stimulation, the animal went back to a reduced conscious state. Eyes closed, the muscle activity disappears, and the resumption of these large slow waves disrupting normal processing. And this was, repeatable time and time again. All right, so basically what I want to do now is, one, yes, we can actually mimic the activity, it seems, of individual neurons in a frequency-specific manner to manipulate consciousness. But I want to show you what this type of high-frequency stimulation, or at least around 40 to 50 hertz, how it influences the rest of the brain. And now we're going to turn back to the mouse model, again, this optogenetic technique I showed you earlier. So what we've got here at top left is a slice through the mouse brain, right? And so here for orientation, here's the cerebral cortex, and here is the central thalamus deep in the brain. So this light green area shows you the cells that have been infected with these light-sensitive proteins, all right? So we can actually control these cells using those blue laser lights. So we're going to put our optic fiber in the brain, and you can see us shining the light on these particular cells to activate these cells, all right? And so we're going to activate the central thalamus now and perform an fMRI experiment, which, if I can actually get here, oops, I have to click out. You're lucky here. Sorry, Kurt. can't see the actual pointer that you're always drawing. Maybe you could show me for one in this video. And
So just reselect the pointer.
oh, that's it.
Deactivate. Yeah.
Oh, too easy. And all right, and I'll take full credit for that. And now I have to come back across the slide, just as a PI would. And all right, so we'll go back to here. Oops, I have to go back this way. Great. So what I'm going to show you now, different slices through the mouse brain from the frontal lobe all the way back to more posterior parts of the brain. And so we're going to be stimulating the central thalamus here using optogenetics. And anytime you see a part of the brain that starts showing a reddish orangey color, that means its activity increases above baseline. So what you're going to see now is we're going to start shining the blue light laser. We're stimulating the central thalamus, and you'll notice the entire brain actually starts to increase its excitability, right? You can see large swathes of the brain that are now activated, which presumably supports that wake up I showed you in the previous slide. So what I'd like to do now, though, is show you the same sort of experimental paradigm, but now stimulating these central thalamus neurons to mimic the actual type of electrical activity they're showing in anesthetized or sleep states, reduced conscious states, right? So we're going to stimulate now at 10 hertz. And when we do this now, what you'll see is that with the same paradigm is the same brain slices, but wherever you see blue parts of the brain, the activity is reduced below baseline. So once again, we're stimulating the central thalamus, but you can see much of the cerebral cortex on the surface of the brain now is actually reducing its excitability. It's becoming deactivated, all right? So the question now, what is the actual consequences of this? Well, now we can actually go back to the monkey model and perform a similar brain stimulation experiment as I showed you before. But now we're going to stimulate at this low frequency of 10 hertz, mimicking the actual thalamic neurons in this reduced conscious state. But we're going to do that whilst the animal is awake and behaving. We're going to try to override the system to basically reduce consciousness of the animal. And that's what we did. So time across the experiment here, we've got various behavioral and neural indicators as before. And prior to stimulating the thalamus, the animal's awakened and behaving. So you can see its eyes moving about, exploring the environment as you'd expect. We had a video of the face, and so we basically translated luminance changes on the videos to these traces here. And so wherever you see deflections, it shows that the animal's face is moving. And not surprisingly, the animal's moving about as well prior to the stimulation of the thalamus. And then in this colored region, we actually turn on the stimulation of the central thalamus at this low frequency. And then we induce what we call vacant perturbed consciousness. The animal goes into a reduced conscious state. All right, so you can see here that suddenly the eyes stop moving. We actually get near complete behavioral arrest. And the animal is just sitting there. I mean, it still has body, posture and levels of arousal. The awareness just seems to change. It's staring vacantly into the distance. And it seems to share a lot of properties with absence epilepsy that you might see clinically. So patients suffering from absence epilepsy, as you may know, will have lapses of consciousness for seconds, tens of seconds at a time. And this can occur up to 100 times a day or more and have a severe impact on their life. So this very much resembles some of the different phenotypes of absence epilepsy, including some of the occasional eyelid flutters we see here that you see in patients, as well as occasional lip movements, as you can see here. All right. So this shows that we can actually bidirectionally manipulate consciousness in a frequency selective manner with this fine-tuned sort of deep brain stimulation, all right? Now, just to be clear, we used a very different approach to what's used clinically now to get these effects. Now they use relatively large electrodes with stimulation through 1 or 2 contacts, which have about 1.25 millimeters in diameter. We used simultaneous stimulation through many small electrode contacts. 12.5 microns in diameter to really try to get at the scale that's maybe more relevant to manipulating individual neurons. Right? And then finally, to sort of summarize why these different stimulations affected consciousness the way they did, we return to our schematic. All right. So what I really want to show you, just for refresh the visual cortex on the left, the parietal cortex on the right, the surface of the brain, the different layers of the brain, and down below the thalamus. So when we stimulate at this high frequency of 50 hertz, we're actually increasing activity in the central thalamus, as shown in yellow. This activates these red pathways here, which then increases the activity of brain cells in those deep layers like layer 5, all right? Basically, then this restores these intracolumnar connections that allow the increased activity in deep layers to increase excitability in the superficial layers. We increase the actual feedback pathways that are, basically carrying information about your attentional priorities and goals. And this increased excitability in the superficial layers then allows you to connect to your environment, to process sensory information from the world around you along these green feed-forward pathways. And then on the flip side, when you stimulate at the low frequency at 10 hertz to mimic this thermic activity of neurons in reduced conscious states, we reduce activity in the central thalamus compared to baseline. We deactivate those pathways from the thalamus to the surface of the brain, the cerebral cortex. We reduce activity, as shown in blue, in the deep layers of the cortex. We then take away the intracolumnar connections that support the excitability of neurons in these superficial layers, which leads to a reduction in these feed-forward pathways, shown here, which now disconnects you from the environment because you can now cannot transmit sensory information, which is likely why macaque monkeys and possibly absent epilepsy patients actually become removed from their environment to have these lapses in consciousness. All right, and now for my final slide. Basically, this brings us to the growing convergence research project that Peter Sieler and Chuck are all working. So one of the limitations of the work that I've shown you so far is that we were using deep brain stimulation at a constant rate, either 50 hertz or 10 hertz. However, the brain, as you can imagine, is much more dynamic than this. So what we're actually currently doing is using machine learning and a closed loop approach to guide deep brain stimulation. So this is where this multidisciplinary team really shines. I'm really speaking about my amazing collaborators here. So we can first take neural recordings from human patients, as you can see here on the right. We can then characterize the neural dynamics in different states of consciousness. We can then model the interactions between neurons in the brain networks that we think are supporting consciousness. We can inform the model by taking recordings now of different parts of the brain in the macaque monkey, because now we can actually target different parts of the brain that weren't accessible to us necessarily in the human patients. We can then now use the model to actually make predictions about the consequences of deep brain stimulation. And then we can then take it into the macaque monkey model again and actually use neural recordings that are taken in real time and behavioral indicators as inputs to our machine learning algorithm, which will output optimal stimulation parameters to increase or at least manipulate the level of consciousness. And the long-term goal then is to bring this back into the clinic. So I would like to thank my amazing collaborators at Stony Brook, and I apologize to many people in Peter Siemer's and Chuck's labs who are not mentioned on this slide. I love you all, and next time I'll use finer, smaller font, everyone else. But in particular, from my lab, I want to highlight Michelle Redenbauer and Mohsen Afrasiabi, who are the principal drivers of all that deep brain stimulation and computational neuroscience work that I showed. So now, questions, please.
Right, questions now?
Please.
What method do you use to try to detect and highlight the brain activity area and show the real life picture? I know in some other biological experiments, they may use the fluorescent protein to do that. I don't know what method they use to do that in the brain.
Yeah, so I relied on like those calcium imaging or fluorescent methods and optogenetics. We're going in that direction to a monkey, but those calcium imaging experiments all in rodents in amazing other labs. So what we use mainly is high density electrode recordings. So we're using these electrodes now known as neuropixels that have 1,000 electrode contacts on each electrode shaft. And so they would implant them in the brain. There's no pain receptors in the brain, so you can implant them, subject's not aware of them, and then measure the voltage changes that occur in the vicinity of each electrode. So we're literally looking at voltage changes in the brain. And because neurons communicate by changing the voltage of their cell membranes, the cell walls, that's what we're actually monitoring, individual neuron changes. Yeah, my pleasure.
Yes.
Chuck, I have a question. So the IIT people are very committed to this like back of the brain idea that it all goes on in sensory cortex.
Yeah.
Is it? I would think that the kind of the core ideas of IIT are kind of anatomy agnostic? Like, is it really, there's something about IAT that means it has to be all kind of sensory cortex, or could these ideas about cause effect structures like involve frontal circuits in principle?
Yeah, I think that's a great comment. And I think proponents of IIT, like Giulio Tononi, for example, would say that, their theory is somewhat agnostic to brain anatomy, and actually argue that it doesn't, as you pointed out, have to actually apply only to organic tissue. It could apply to inorganic sort of circuits or whatever else as well. And so part of the reason why the proponents of integrated information theory, like Giulio, Christoph Koch, suggesting it might be more of the back of the brain contributing to consciousness is that there's for example, some of the data that I showed you, obviously like electrical stimulation effects, but on a more theoretical grounds, there's a paper by Andrew Horn, a postdoc in Giulio's lab, and on the slide. And Julia published a paper, I think it was in the journal Entropy, where they looked at the sort of types of characteristics of brain structures that might support our particular type of extended visual experience. And they were suggesting that grid-like structures that you find in some of the more posterior parts of the brain might be more apt to representing the world around us in this extended format. So they made arguments based on this link between what they call conceptual structures that represent the contents of consciousness and these grid-like patterns of activity in more posterior parts of the brain. But I agree, it doesn't necessarily have to be in the back part of the brain, but it does make for more interesting debates when there's conflict. A neuroscientific soap opera, so to speak.
Thank you.
My pleasure. Please.
For your optogenetics experiments, how are you influencing the frequency of central thalamus activity? Are you just making pulses of light at 10 hertz, for example, and that is enough to?
Yeah, and I should be clear, the data I showed you is not from my lab. I wish it was. It's beautiful data. So data, for example, from Matthew Larkin's lab, which was published in Cell. That was the study where they're optically stimulating the dendrites and then measuring the coupling between dendrites and the soma of the layer 5 pyramidals. And they're literally pulsing the laser at the same frequency that they're trying to induce of individual cell activity. Because you can imagine, switching the actual ion channels on and off, so to speak. At least that's the idea.
Is there like an established knowledge on the resolution to which you can turn channels on and off? Like, you might not know this.
How fast you can drive them?
Yeah.
I think it depends a little bit on the options. And Asima may even know more about this being in Mike's lab at one point, Mike Klass's lab, who does these techniques. But yeah, I think it depends on the options as well. But I don't know enough. There's even some talks that I saw recently at a conference where the effects are probably much more complex than what's originally suggested. They can get changes in gap junctions, so electrical coupling between cells, as well with these techniques. So the effects are difficult, but nonetheless, if you record spikes, which I think are on the part of the slide that I flipped over very quickly. I think I flipped over it again, did I? Maybe it's one back, sorry. Easier just to go back. There you go. You can see that you get the changes in the bottom right slide in spiking. And it's probably not easy to see the dynamics of the changes in spiking there, but it does seem to follow the frequency. Seema, do you have anything to say? I know it's not exactly a field, but I thought.
No, but I was thinking about the model that you're building.
Yeah.
I mean, so what we have in the model, we have these node cells in the superficial, and these node is superficial and deep layers. And here, like looking at those, like the cells that that the dendrites and so on. What do you think, like? So one possibility is to, instead of modeling these cells as these nodes, you model the dendrites and more sophisticated way of like having the whole neuron like from superficial extending to the deep layer.
Yeah.
What we are doing, we have one node in superficial, one node in the deep layer. Do you think is it necessary to switch to a more sophisticated model that we have one neuron?
It's a great question, and I guess the whether it's easily implemented is more in your realms that I can comment. But the reason why I was happy with having the superficial and deep distinction is I thought it captured not only this framework, but also the neurons that are in superficial, like cell bodies and superficial layers and deep layers, which have more local connections from superficial to deep. I thought we could capture both.
Yeah.
And I think the role of the superficial cells and the deep layer cells are probably having different functions. And so to be explored more with that data, and that's fine. But I think if it can be modeled, then great, let's try it. We could have these layer 5 cells and then the superficial cells modeled separately. I think it'd be amazing, especially with a task like the rule task where you've got like easily sort of specifiable contents of consciousness. Yeah. And then you could see the different roles.
I agree. I think for the scope of what we are looking at, maybe the current model is enough, but in the future maybe.
Yeah, and it's important to note too that the central thalamus and medial dorsal thalamus have different patterns of connectivity too, right? I mean, the medial dorsal thalamus will tend to go more to the middle and superficial layers, right? Although there are some weaker connections to deep layers. So there's a lot of complexity here. I think you've already got an incredibly complex model, which is doing amazing things, coming together, converging with the monkey data that we're seeing. So, I mean, but yeah, if we can get it even more refined, that would be amazing. And that's why I chose to work with you guys.
We are lucky ones. Thank you so much.
Question. On memory and thinking because the stimulation part of the consciousness.
Yeah.
Because all of these experiments are based on external, all of the
pretty much, except for maybe if you mean the initial binocular rivalry, does that count as external stimulation too? That was the presenting different stimuli to the eyes. Oh, it's okay. Yeah, I guess internal stimulation is what we focused on because it was something that we could test our theories directly by going into brain areas and have a very clear change in consciousness. Like in the monkey, it's very difficult to know exactly what they're perceiving, although you can train them to do the binocular rivalry task, for example. And so when you have something that's easily detectable as waking up or being asleep, for example, or anesthetized, it makes the changes quite clear and shows that things are really working if you can really wake the animal up. But you're right, we didn't explicitly test contents of consciousness in these tasks. What I can say in data that we're currently preparing for publication, we'll hopefully submit it sometime next month. We had an auditory audible task in this particular paradigm. And now I'm going to embarrass myself by making weird sounds. So this is a task where you present a number of tones, standard tones like pop, 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 followed by an audible, an unusual frequency, pop. And so when the animal hears it, pop, 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 and the idea is usually there's some sort of prediction error signaling coming from the brain when you hear this audible tone. And so what we're seeing is that during anesthesia, that response to the audible was no difference to standards. But during wakefulness, as some others have shown, using not this paradigm, but when you compare wake to sleep, for example, the response is much larger to that audible stimulus, which suggests that you've got a neural representation of the content of consciousness in most cases. So we're trying to get at those questions, but they become more difficult. And unfortunately, none of my reviewers at major journals would believe that I'm the only one who can mind meld with a monkey to read their monkey. Yeah. Does that help answer your question?
That's fine. I was just wondering if the model would be different if you're just doing deep thinking and no stimulus.
I mean, without electrical stimulation.
Yeah, no stimulus.
Yeah, we did comparisons. I didn't show you the whole data because of time, but just comparing awake versus sleep versus anesthesia. But again, I agree with you 100%. We haven't really gotten into the contents of consciousness, but I'm involved, for those who are interested, in another major project through the Templeton Foundation, where we're doing just that. We're actually presenting a behavioral task where we're manipulating exactly what the monkey sees and recording from all across the monkey brain to get at the contents of questions. So our project that we're all working on is really getting at, understanding the neural correlates of consciousness and manipulating the actual, neural activity in a purposeful way. These type of experiments where we have the monkey doing a task and reporting what they're seeing is getting more of the content, I guess, without the brain stimulation. Does that make sense? Yeah. Although we will perturb the frontal lobe in these experiments to see if the monkey still perceives stimulated state.
Questions?
May I have a couple of questions, general questions?
Please.
Yeah, so basically, question one, do you know the difference between the power consumption of brain during this deep sleep and conscious state? And the question two, how do you actually can measure power consumption of the brain?
That's a great question, Mikhail. You're breaking up. I didn't hear those questions. I'll see you later. No, I'm kidding. The energy consumption, if I can go down that route, can be monitored using, I guess, glucose metabolism. You can do that using techniques such as positron emission tomography, or PET. The idea here is you inject radioactive nuclides that aren't harmful into the subject. They're going to emit positrons as they flow through the brain, which will react with nearby electrons, give off gamma radiation, which you can pick up with external detectors and see where these signals are coming from. And you can use techniques like that with glucose isotopes, which can give you an idea of the energy metabolism of the brain. If my limited understanding suggests that certain parts of the brain does drop during reduced conscious states, I don't do any of the work there. But there are a number of different changes that occur. The actual number of synapses in the brain, the connections between neurons, seems to be scaled down at different points of sleep, as well as the strength of those connections too. So I think generally speaking, I'd feel comfortable saying the energy metabolism goes down in reduced conscious states, but sadly, it's not in my area of expertise. Does that stop? Oh, I can see you, Mika.
Yeah, finally found. And question one was, what roughly the power consumption? 20 watts. I read this number like in some papers, 20 watts. But is it correct?
That is a great question. I'm going to say yes. No, I have no idea, but it's a great assumption. I'll look into that and I'll feed the answer back through Petra to you, hopefully. But
okay, thank you.
I'd like to know that answer now, especially when I give my next talk in an engineering department and then start asking very clear number questions, which I just hand wave them.
Any other questions from the Zoom box? I got a question, Yuri, about the experiments you had with the thalamus stimulation. So you had two slightly different responses. 50 hertz is one type, and 10 hertz is a different one. So what is actually physiologically happening with the thalamus that it responds in so different way?
That's a great question. So, we in our setup aren't able to record the activity simultaneously as stimulating. We don't have a switching head stage, as it's called, with our particular experimental setup. So I don't know in our specific case, but what I do have is from other subjects, sort of work using stimulation methods in animal models. And they suggest, for example, that indeed, sometimes they're using much larger electrodes, so that might be hard to change. But if you stimulated 200 hertz, much higher, which we actually did as well, which we actually induced the same lapses in consciousness, you get an initial burst in activity followed by suppression of activity. In 10 hertz, you don't necessarily seem to get that same initial burst, but you do get a reduction in activity. With 50 hertz, it's a bit hard now because there's a little bit of heterogeneity in the different subsets of neurons in the central thalamus. So I think with these smaller cases, based on the fact that we get similar results, at least with other labs using optogenetics, it increases the spiking activity, somewhere in the vicinity of the frequency that we're stimulating at. I don't know what's really happening clinically with these larger electrodes. That's much debated as we had in our review paper. I'm going to sort of bring as many convergent ideas into the one answer. But so I don't have a direct answer, but indirectly, I think the neurons are actually spiking, at that level that we're stimulating until we get to very high frequencies when initial burst and things shut down. And that likely is due to specific protein channels, calcium channels, that likely are inactivated after initial burst of stimulation.
One more question. In your opinion, it's kind of speculative. What kind of role do you think this research on consciousness will play for the growth of artificial intelligence?
Yeah, so just a really easy, short question. So I think the singularity is nigh, run for the hills. No. No, I think that it gives us an interesting insight into types of structures, whether organic or not, which might be able to support consciousness. So I think that's one insight here. Beyond that, I mean, it might help with designing, if one were to do that, artificial intelligence systems that might be able to give rise to conscious experiences. So one thing we haven't sort of dwelt with here, and it's going to get a little bit hand wavy, is that we haven't talked about the self-consciousness aspect, that you need a representation of self, so memories of self somewhere. And so, you know, if you're designing a system, you might have to have some part of that artificial intelligence system that has memories of itself, somehow, you know, encoding its position relative to everything else, which then it can compare incoming sensory or other inputs to sort of, you know, compute changes from its current state that might reflect, you know, perceptual experiences. And so I think that this sort of work, as it relates to the artificial intelligence, it really might narrow down the parameter space of what type of, you know, circuit designs or other designs might actually be able to support consciousness. I'm sorry, it's a bit hand-wavy, but that's the best I can do at this time. And the machines are ready to swing.
That's a goal for year 5.
Absolutely.
Maybe the next project.
No, it's a great question. I mean, your insights, I mean, you've clearly thought about this.
Westworld, that people follow not people who are actually computers. No, I don't know, difficult.
Yeah, but I think it, you know, it does suggest that, you know, in many of these theories, one of the key attributes is having reciprocal interactions between computing elements, whether they're neurons or something else. And so that would have to be a critical aspect of the design, to have these sort of feedback loops built in, which allow resonance, you know, in these different circuits, whether it's in the brain or organic structures.
It's kind of unnerving when for those of us who work, say, on machine learning problems, we can solve any difficult problems. And what is not pleasant is that the computer doesn't know what it's doing, actually. It's just doing it.
Yeah, or at least it's not telling us what it's doing.
Yes.
I think, Paolo, did you have a question online?
Yeah, I have a question for what you presented today. I understood that when we are in a wake condition, we can observe that we have this 50 Hertz activity of the brain and different parts of the brain, while we have something closer to 10 Hertz when we are in a coma or not so awake condition. And so this motivated you to stimulate at 50 Hertz to increase the awareness of the subject. So the question is, do you think that the presence of this 50 Hertz is just, let's say, a condition to let the brain communicate much faster, or is exactly the communication from one part of the brain to another part of the brain that happens at this speed, let's say at this higher speed when you are in a awake condition? This is the first question. The second one is
You're breaking up. I'm kidding. Go.
and okay, let's stay with this one. Otherwise, it's too much.
I should first say that Paolo and all our labs have been Zooming for a couple of years, and I didn't have the sort of time to sort of bring this up, but Paolo, like me, is astutely challenged, and then suddenly started wearing black, thick polyethylene frame glasses in the last few weeks, and it was like staring in a mirror. And you're not wearing them today, so I wasn't sure if it was a gag or not. But anyway, I think in your question, I think the part A or first option, perfect. I was like looking at myself on screen. But your Italian is better than mine. The first option, I think by stimulating at 50 Hertz, and it could probably be 40 Hertz, and clinically they might stimulate, like Nico Schiff's group, close to 100 Hertz, might be just sufficient to increase the excitability in the cortex, you know, for example. So I don't think there's something special necessary about 50 Hertz in itself, if that's the question. Is it transmitting information? These circuits have been, with the central thalamus, suggested to be involved in, for example, guiding eye movements and attentional priorities. So it could carry additional information to that, but I haven't really been able to probe those questions yet. It's something that's on the agenda you know, on the long list of experiments that we all want to do in the future. Does that kind of get at that first question you were going to ask, Paolo? And the second part that you hadn't asked yet?
Okay, and do you have any idea which part of the brain generates this new frequency? I mean, it could be something like, let's say, what you have in the heart, you have pacemaker cells that gives the rhythm to all the others. I mean, such that you have kind of a switch in the brain that lets you operate in one modality or the other one.
Yeah, so great question. So if you look at the slides now that I'm showing, so this is the medial view of the brain, right, human brain. And the part that we were stimulating is the central thalamus, shown here by ILM, but it receives information from the brainstem, what's called the reticular activating system. So you can see these pathways that come up to the central thalamus as well as the cortex. If you actually damage those brainstem structures, you have you know, a coma state typically, right? And so it seems like you necessarily need activation from the brainstem to actually support certain levels of excitability in the central thalamus and the cortex. But beyond that, I think there's reciprocal connections between the central thalamus and the deep layers of the cortex, you know, help to sustain activity in both regions, like some sort of, you know, to use a little bit of a corny sort of metaphor, like an engine of consciousness, you know. And so if you have spiking in the central thalamus, that's going to excite neurons in the deep layers, which in turn will excite the thalamus, and they get into these sort of resonant activity patterns. And I think that's what sustains, let's say, that baseline level of activity, if that makes sense. So, you know, I think there are, I don't want to give the impression that just one brain area like the central thalamus that is helpful here. I think there are multiple different parts of the brain that contribute, maybe differentially, but particularly for manipulating the level of consciousness, the reticular activating system in the brainstem here, the central thalamus that we were stimulating, and the deep layers of the cerebral cortex on the surface of the brain all seem to be necessary, and not just those. Some of these other deep brain areas here, for example, seem to also interact with these structures. So yeah, it gets more and more complex the more you look.
Okay. Thank you,
I
Chad.
wish I could give you a simple answer. I'm just going to say this part. No, I'm kidding. Yeah.
Actually, I do have a question. You're talking about the thalamus, and it gives frequencies of 10 hertz up to 50 hertz.
Yeah.
I was just wondering if you've noticed any difference in behavior. Look at it, if you're like at 20 hertz or 35 hertz, what happens?
Yeah, it's something that we'd love to do, and hopefully we'll get to explore with this you know, machine learning guided stimulation. You know, these experiments were 12-hour long anesthesia sections, so it's a very long day, and you're gowned up for the entire time and not eating. For me, not drinking caffeine. I mean, sweats and itching for that period of time, and so basically, you know, the idea was to try to span as many different frequencies that were physiologically relevant, and some of those used clinically. So we just used 2, 10, 50, and 200 hertz for that range, and then reproducing enough time so we could do the appropriate statistical modeling. But it's something like, yeah, we'd love to look into, you know, see what happens at different rates.
Yeah.
50 hertz might not be optimal, right? I mean, talking to Nico Schiff, you know, and he has views that maybe at higher frequencies might be better, at least with his clinical patients. So yeah, it's something we'd love to explore, but it's such a large parameter space. Right? You can change the current, you can change the frequency.
I'm kind of surprised you've gotten as far as you have.
Me too, but that's ripped out all my hair, and but Australians are fearless. Yeah.
More questions? Well, Yuri, thank you so much again.
See you all. Thanks for the questions. Good to see you, Paolo. And I'll get back to you, Mikhail, about 20 watts. I'll experiment with car batteries.
Thank you, Yuri, for your presentation.